Good morning, everyone. So this session is signaling. We will have 8 speakers. May I remind all speakers, you have 12 minutes for talk, another 3 minutes for discussion. So the first speaker will be Aaron Savage from University of Sheffield. He's going to talk about tmem33 in angiogenesis. Please.
Hi. First of all, I'd like to thank the organisers for the opportunity to speak here. I'm a PhD student working with Rob Wilkinson and Tim Chico, and I'm going to talk about my work on calcium signalling during angiogenesis. So, 2 of the leading causes of death worldwide are cardiovascular disease and cancer. In cancer, we have excessive angiogenesis, which leads to tumour growth and is ultimately the main route of metastasis. Conversely, in cardiovascular disease, we have a blocked artery and insufficient angiogenesis to revascularize around. Current treatments have been largely unsuccessful using VEGF, so the identification of new targets to modulate angiogenesis in these cases is essential. So angiogenesis is the process of forming new blood vessels from those which already exist. So endothelial cells are specialized epithelial cells lining the lumen of blood vessels. In response to VEGF, which binds to VEGF receptor 2, tip cells begin to migrate from the endothelium and lead the new angiogenic sprout. As the cells migrate, DLL4 is upregulated, which induces notch signaling in neighboring cells. This downregulates VEGF receptor 2 expression and upregulates VEGF receptor 1, which is unresponsive to VEGF. This means that the cell following the tip cell, called the stalk cell, can't respond to VEGF and is reliant on the migratory tip cell's response to VEGF for migration. We know that calcium signaling lies downstream of VEGF signaling, but we don't know its role, and that's what I'm trying to find out. Endothelial cells are non-excitable cells, which means they don't use voltage-gated calcium signaling. Instead, they use store-operator calcium entry, which is when the endothelium, the endoplasmic reticulum, which is the major store of cellular calcium, becomes depleted. The cell acts to bring calcium back in via different channels. One well-characterized channel is the calcium release activated calcium channel, made up of STIM1 in the ER membrane and RI1 in the cell membrane. And other channels known to bring calcium back into the cell are TRP channels such as PKD2 and TRPV4. So we're interested in endothelial calcium signaling. So we generated an endothelial reporter of calcium signaling, which is very similar to the one recently published by the Kawakami group. So we use GCAMP, which is a circularly permuted GFP, which in the presence of calcium undergoes a conformational change, leading to increased GFP fluorescence. So we can see changes in real time, and here we can see this in the video. The tip cell just here is about to divide here, so it's just undergone cell division and is now beginning calcium signaling as it migrates again. As I said before, calcium signaling lies downstream of VEGF, and we can show this using this line. So here we can see in control embryos, we get a change in GFP fluorescence over time, which corresponds to changes in calcium signaling. When we inhibit VEGF, we lose calcium dynamics, and we also halt migration entirely. Conversely, if we inhibit notch signaling, which is an inhibitor of the VEGF response, we increase calcium signaling both in frequency and amplitude, which fits nicely with what's already known. So where does tmem33 fit into this? Well, it's a 3 transmembrane domain found in the ER membrane. As you can see here, it co-localizes with PDI, which is an ER membrane protein. And as we said before, the ER is the main store of calcium. So we know that it interacts also from work with our collaborators with PKD1 and PKD2, which can form a calcium channel. So we think that it's involved with calcium signaling. So we thought we'd have a look at where it's expressed in a zebrafish, and we can see that it's expressed in both the endothelium and the pernephros, the developing kidney. If we inject tmem33 morpholinos, first of all, we get delayed migration, which you can see in panel D compared to C. We get impaired arterial venous specification in panels E and F, and we get incomplete anastomosis in G and H, we can see in H, and aberrant branching patterns. And we show, in panel I, it shows that the splice blocker morpholino works. So as it's expressed in the kidney, we had a look in a kidney reporter line. So you can see in the center of the... Oh, hang on. Sorry. The shape here, like this in the kidney, is the glomerulus. That's the word. Is the glomerulus, and if we inject the morpholino, we get increased glomerulus size corresponding to kidney cysts, which is what you see when you knock down PKD2 as well. So I injected this into the calcium reporter line, and we see an attenuation of calcium signaling on the right, which is very similar to when we inhibit VEGF signaling, and we get delayed migration. So it also looks as though the endothelial tip cells are kind of flattened and blunt. They seem to be lacking filopodia. So we generated a filopodia, F-actin reporter line, which labels filopodia in the endothelium. So you can see at the control on the left, we see filopodia extending as the cells migrate, whereas on the right, the morphin lacks filopodia and has delayed migration, which I've quantified in panels E and F. So we see in E, we see a reduced number of filopodia, and F, we see the length in micrometers of cellular extension, indicating that we retain lamellipodia, which account for the small membrane movements, but lose filopodia. So at the moment, we're generating quite an interesting story. We've got an interesting phenotype. So we thought we'd make a mutant. But as I'm sure many of you have experienced, the mutant had no phenotype, which was a bit annoying. So we thought it was the end of the project, pretty much. So we got a 2 base pair deletion, leading to a frame shift, causing a truncation in exon 3, which is in the middle of the conserved 3 transmembrane domain. And if we look at the expression levels in both wild type on the left and homozygous mutant on the right, we get nonsense mediated decay and a loss of expression, or decrease in expression at the very least. But the homozygous mutants are completely viable and grow with normal vascular patterning. So we thought this was, we're on the verge of shelving this project when the paper from the Stanier group came out suggesting that many mutants display compensation. So you test this by injecting the morpholino into the mutant. So here I've done a heterozygous incross of tmem33 fish and ranked them for severity of phenotype and retrospectively genotyped them to find out what the genotype was. So on the left we have tmem33 wild type, in the middle we have het, and on the right we have mutants. And you can see that in the wild type, over 50% of embryos display severe defects, while very few display no defects at all, while the opposite is true of mutants, indicating that there's a protection against the phenotype and suggesting we're seeing some sort of compensation taking place. So we repeated this with the kidney and found the same thing, that the mutants also display protection against the phenotype. So we think that something is compensating, but we now want to find out what is compensating. So we're planning on doing an RNA-seq, but first we decided to kind of validate the knockdown phenotype by using a different technique. So we used CRISPR interference. So in a normal CRISPR-Cas9 system, you get site-specific mutations guided by the guide RNA, and you get a mutation setting off homologous repair. So with dCas9, which you use in CRISPR interference, it lacks the endonuclease activity. So it kind of sits on the DNA and acts as a transcriptional block, and RNA polymerase comes along and can't transcribe. So we're making great use of this. I designed some guide RNAs for tmem33 and a control guide, and injected these along with dCas9 mRNA. We found that the expression is decreased in the knockdown, and we get vascular branching defects as well as cystic kidneys, which I've quantified on the right there. You can see that we've got tmem33 in red in the graphs. We see increased vascular phenotype, and then at the bottom we see an increased glomerulus area. So I decided to try this again in the calcium reporter line. So I injected this in, and again, this recapitulates the morphant phenotype. We lose, we get attenuation of calcium signaling, and delayed migration. So I thought I'd have another look again and stick it in the filopodia reporter line. And again, we recapitulate this. We lose filopodia. We get lamellipodia and delayed migration. This embryo here has got some severe branching defects taking place. But as I said before, there are some problems with ubiquitous knockdown. We know that what we could be seeing is an off-target effect. So I've decided to try and do some tissue-specific knockdown. So I'm using the endothelial promoter to drive DCAS9. And this is what we see. So the construct is a TOL2 construct. I've just said before, all of this is transient experiments. We're currently growing the line up to use a stable line. So just to recap, I've used the figures from before. The first panel underneath, in fact, no, sorry. The construct itself has got a fly-1 endothelial promoter driving DCAS9, and then using a CRISPR, which is blue eyes, as a positive marker for transgenesis. So we can see which ones have got blue eyes, and then we know that they're transgenic. So to recap, using the control guide RNA and ubiquitous DCAS9, we get neither vascular nor kidney defects in panel A and D. Using ubiquitous DCAS9 and tmem33 guide RNA, we get both vascular and kidney defects. But using the tissue-specific DCAS9 with the tmem33 guide RNA, we get only the vascular defects, and the kidney is normal, which we can see in the quantification on the side. So we retain the vascular defects in the graph, and then the graph below shows that we lose the kidney defects and it returns to the control area. So this suggests that the vascular defect is cell autonomous, and it also means that we can now start to uncouple both the kidney and endothelial phenotypes. We can perform tissue-specific knockdown and see whether different genes are compensating in different tissues, and this is something we're looking to do in the future. The kidney work is kind of on the side at the moment, but I'm more interested in the vascular work, but so that's why I focused on it for this talk. So that brings me to the model. So we think in the vasculature, we think that following VEGF signaling, we get tmem33 mediated calcium signaling, which is essential for filopodia motility and even possibly F-actin polymerization. Filopodia are rich in VEGF receptors, so it means that as well as being a mechanical process to move the blood vessel, they also sense VEGF and help migration and guidance. Whereas if we lose tmem33, we lose this, and this accounts for the delayed migration and misbranching. So with that, I'd like to thank my supervisors, Rob and Tim, our labs. Jane's just here. She's got a poster and a talk she's going to see tomorrow. Everyone else in the lab, the Frakes group, and our collaborators in Nice. Thank you. I'm happy to answer any questions.
Hi.
Hi.
Nice talk. Until you figure out why your mutants are being compensated, it might be that they're still sensitized. I wonder if you've tried, VEGF inhibitors or Notch inhibitors to see if, do a dose response compared to wild type. You might find that, in fact, they're sensitive.
That's a great idea, actually. Yeah, thank you. I'll try that. Thank you.
Hi, thanks for the talk. I was wondering about your podocyte phenotype. Do you know if do you have any idea about what causes the phenotype in that?
The loss of filopodia?
Yeah, but is it also dependent on VEGF?
I don't know. That's something we've not looked at the moment, but I don't think from the literature it would be. But I think the calcium signaling kind of is required for filopodia and F-actin mobility. So that's kind of what we were thinking. Okay, thank you.
Hi, I was curious if your tmem33 mutants were your homozygous mutants were maternal zygotic loss of function, or were they zygotic?
Oh, yeah, they're maternal zygotic, yeah.
Okay.
Yeah, we tested it, yeah. Okay, thank you.